Now let us hear Professor Ivo Bianchi. He is a dear friend. I have known him for many years. He was a lieutenant doctor in prison many years ago when I had occasion to meet him. And we're talking about the central role of the mitochondrion in organic homeostasis. He is fixated by the mitochondrion and always has been, and he will be highlighting how that there is the duality between mitochondrion and cell is not in opposition. Thank you, Steno. Yes, I only talked about mitochondria in the last two meetings, and I now have to talk about the cell. But above all, I have to talk about the correct medical approach, not so much to killing the cancer, but to reach an equilibrium. Health is equilibrium. Let us see how pathologies are often have an anatomical, genetic and intrinsic basis, whereas in terms of intoxication, then disease is called mitochondrial medicine. As I said, in terms of to have an anti-aging effect, both approaches are required. Here we see two great scientists I cited before. Otto Warburg, who talked about the importance of metabolism and the role of toxins. But we mustn't forget the other major Nobel Prize winner, Frederick Sanger, who was awarded 2 Nobel Prizes for studying molecular genetics, studying DNA and endogenous factors.
Diciamo 2 parole ancora sul mitocondrio.
So, at the last 2 meetings I talked about mitochondria as extremely important in producing energy for all our cells. All cells contain 300 to 400 mitochondria, but if it is more active, then it contains more mitochondria and adapts itself, and mitochondrion adapts itself in shape to the cell's activity. To understand the importance of mitochondria, 10% of the weight of our organism is made up of mitochondria. The functions of mitochondria are to produce energy, and they produce energy without producing waste. Mitochondria can break down their own waste, producing carbon dioxide and water, so it has a role of energy production and purification. In addition, mitochondria regulate the body's functions by deciding when a cell is old, when it is not functioning, when it is unable to work properly, and when it is working as a tumor cell. So mitochondria regulates and decides on apoptosis. It decides when cells have to die. We will also see this later, to see whether there is an excess of mitochondrial activity. We will have an excess loss of cells, whereas when mitochondria are under-functioning, we will have a proliferation of cells. When we have calcium cations in the cell, we have problems, and mitochondria will regulate membrane functions and control contractions of the heart, and therefore mitochondria have an extremely important function in the myocardium. Again, the mitochondrion is underestimated because it captures cholesterol, and from cholesterol synthesizes steroid hormones. So all the sex hormones, all the hormones involved in stress, and extremely important vitamin D, and therefore plays an extremely important role in metabolic regulation. So virtually everything depends on the mitochondria: energy, sexuality, fertility, and the survival of the body as a whole. Here we can see a scheme of how the various substances from food intake are processed by the mitochondria to produce energy in the form of ATP, regulating cell functions and producing amino acids and glucides depending on the cell's needs. So it receives generic amino acids and processes them as the cell needs them. So very many of these cells' needs today are in fact have their basis in mitochondrial function. So keeping mitochondria working properly means preventing myriad diseases. We also at this point need to introduce another concept which we can attribute to this great American scientist Lynn Margulis, who developed the theory of Darwin's theory of endobiosis, that a struggle between living beings is responsible for evolution, but Margulis said that the symbiosis, an important component to evolution, is not just due to this, but it's an important question of interdependence and interaction among organisms. So there is not just a nuclear proliferative factor, but also a mitochondrial energetic factor. So these two, life on Earth was able to develop thanks to this mutual symbiosis, because the cell tended to proliferate primitively and led to energy production, as you can see from this diagram. By providing energy and giving the cell potential, it led to evolution. So from the primordial waters on the earth, life was born. On the one hand, we have the protobacterium, from which the mitochondrium will originate, and subsequently we have the archeobacterium, from which the cell will originate. This is very important to understand the cell and the mitochondrion. It's important to understand what they are and how these archeobacteria and protobacteria work. This is a sort of unitary origin. There is the first element, which is hydrogen and glycine, and then a first cell, an anaboid cell, an asexual cell, but then evolution then imposed bifurcation with a propulsive dynamic equilibrium. And this came about by dynamic progression. So I've also introduced the theory of a dynamic balance, according to which life flows not in a linear, stable linear fashion, but in an oscillating progression made up of ongoing adjustments. Here, going back to the archeobacteria, which are the precursors of the eukaryote cell, depend on a continuous supply of hydrogen and carbon dioxide, and produced a large quantity of methane, and these were bacteria, which were life forms unable to adapt, or would have been unable to adapt, to the fact that the primordial earth saw an increasing amount of oxygen produced by algae. So these life forms suffered from a progressive loss of hydrogen ions. This archeobacterial cell was unable to use oxygen, which was toxic for it, but produced energy through glycolysis, which Professor Canestrari talked about earlier. This is a very primitive production of energy and also tend to produce lactate and alcohol. But the eukaryote cell has written in it all the programs of life. So it supplied the structural programs, the know-how whereby life could evolve from the small DNA of the mitochondrion. Thanks to the extremely complex DNA protected by the double helix of DNA, life was able to evolve. On the other hand, these protobacteria, similar to Rickettsia, studying them today, we see that they contain a large quantity of the CoQ10 enzyme, which offers more proof that mitochondria derived from these protobacteria. Another feature of the protobacteria is their great adaptability. They were able to live in conditions without oxygen or very rich in oxygen at very high temperatures and at high salt concentrations. So the mutual symbiosis that the cell has had with these bacteria has allowed the cell to overcome these major changes that were taking place in the oceans and in the atmosphere over the space of thousands of years. Protobacteria can derive from the mitochondrion the ability to use oxygen. They have catabolic and energetic characteristics. They can produce hydrogen and carbon dioxide from oxidative phosphorylases, i.e. Krebs cycle, and also produce water by consuming oxygen. Here we see that these protobacteria are something like molds. They cannot directly use inorganic substances found in nature, but to survive they must use eutrophic organisms, which are of course the archeobacteria, able to directly use inorganic substances. Here we see the protobacterium and the mitochondrion using oxygen to produce hydrogen and carbon dioxide and water, and on the other side we see the archeobacterium and the primitive cell utilizing glucose, H2, which is hydrogen supplied by the mitochondrion, and carbon dioxide to produce lactic acid, alcohol and methane. So we need both oxygen ions, and we also need oxygen, ionic oxygen, for the needs of the mitochondrion and the needs of the cell. Here we need to bear in mind that the cell decides on cell death, which is activated by enzymes, caspases, which are activated, leading to cell death, which can be advantageous to prevent diseases like cancer. Cancer is a consequence of a hyperactivity of this caspase enzyme, but if the caspase is overactive, there will be an excessive cell death, and we will have progeria, this genetic disease which leads to early aging, as you can see in the slide at the top in the photo. And the other situation will be the elderly person, where there is a predominance of anabolic cell function with a relative reduction in mitochondrial function. Here we see from the formulae on the slide, we see from the basic molecule of cell food is extremely important, because here we have a scission. When cell food is placed in water, we have a sulfur residue and deuterium hydroxide, which in turn is split into hydrogen ions and oxygen. This means that when we supply deuterium sulfate, we are not merely supplying oxygen, we are oxygenating the cell. We are not only stimulating mitochondrial function, but we are also supplying hydrogen for cells, because we also have to bear in mind this balance, because it's not simply a question of the cell and mitochondrium, but the mitochondrial balance. We have mitochondrions producing steroid functions and cells which are responsible for their own activities. So all of these, both are required with deuterium sulfate. Then Deutrosulfazyme is the content of cell food, which is a mixture of 78 trace elements, not just Deutrosulfazyme, 34 enzymes and 17 amino acids. So this is an extremely complex drug. I think this is important because our concept and what we need to promote is not just mitochondrial function. Often in medicine we talk about stimulating and suppressing, but it is very important to act carefully on mitochondria, and this emerges from what happens in nature. We see that those who are using the best of mitochondrial function are birds. And this was an important point of reflection. You see that birds at cell level have many mitochondria, and they also have many respiratory chains. So they have a great mitochondrial capacity, which gives them a large energy production, and they also have a low production of free radicals. So a high production of mitochondrial cells leads to a low production of free radicals, whereas mammals have few mitochondria, few respiratory chains, so to be able to produce energy, they produce a large number of free radicals. This is the idea. Then, of course, there are differentiations between mammals and birds, because the larger the animal, the lower the metabolism, and fewer free radicals are produced. And that means we can understand why a mouse lives for a year and an elephant lives for 150 years, and why an ostrich will live for 80 years. So everything's relative and is all to do with the speed of metabolism, which is linked to the number of free radicals that are produced. For this reason, in birds, as there is a high number of mitochondria, there is a very low incidence of cancer, because their mitochondria are not destroyed by hyperfunction, whereas in mammals have a very small reserve of mitochondria, and the consumption of complex 1 in the respiratory chain is very high, so we have a very high incidence of degenerative disease and cancer. What does Deutrosulfazyme do? It improves mitochondrial efficiency by activation, which reduces the production of free radicals, not so much stimulating mitochondria, but reducing the production of free radicals. Because were we to stimulate mitochondria alone, by having a person breathe oxygen continuously, the person would have more energy, would feel highly stimulated. For example, I've heard that at certain parties they use oxygen cylinders to breathe in gas to feel more efficient in terms of brain activity. So stimulating mitochondria will produce more energy, but it will lead to the production of many more free radicals and will lead to the destruction of brain cells very quickly and lead to Alzheimer's very quickly. So the idea of the protective effect of mitochondria is extremely important, especially for mammals, and man in general, but especially for athletes, because they are subject to fast metabolic, accelerated metabolic production, leading to the production of free radicals, as I said before. So it's important to give energy, but also protect against metabolic damage from an excess of energy. So cell food is definitely a mitochondrial stimulant. This is already established. Today we've seen that it also is an anti-neoplastic protection, because an active mitochondrion will understand when the cells are being transformed into neoplastic cells and lead to apoptosis. So the more we stimulate mitochondria, the more we protect against cancer, but at the same time, cell food also gives us hydrogen ions. So it also has this antioxidant function, which will avoid tipping the balance in favor of Alzheimer's. So instead of going to the other extreme, the hydrogen ions will have an anti-degenerative protective effect, to have a balanced role of hydrogen and oxygen, so that efficiency and longevity, which is our target, we don't want to live longer, but in a comatose state we want to be more efficient and live longer. So in terms of treatment, this is extremely balanced. When we talk about amino acid complexes and a complex of the periodic table, an enzyme complex, and then the two key elements of life, hydrogen and oxygen, we therefore have a guarantee of true health and true prevention against disease. Thank you very much.
Grazie a te, Ivo, perché ogni volta che si parla.
Thank you so much, because whenever you talk about mitochondria, we're fascinated and understand the importance of mitochondria in cells by studying the forms of life that they derive from and the use of cell food that provides nutrition to cells and mitochondria.